Hello students, this is Dr. Faizan Mirza. We are discussing selective reabsorption. So, in excretion, we know that when the glomerular filtrate is formed in the Bowman's space, it is having all the waste products which were in plasma that adds up into the glomerular filtrate. But together with it, even the important ions and the nutrients and a lot of required water is also there. And you can't allow everything to go out in the urine, which is in the filtrate right now. So you need to reabsorb those things back. So the solutes which are required by the body, they need to be reabsorbed back into the blood so they don't end up being sent out in urine. And only the waste products and the excess of salts and water and metabolites of drugs and hormones, they go out. So how selective reabsorption occurs? Selective reabsorption occurs mainly all along the nephron, but primarily we are discussing the proximal convoluted tubule. The proximal convoluted tubule accounts for about 67% of selective reabsorption, which makes it the most important region allowing reabsorption to occur. It is called proximal convoluted tubule because it is near the origin of nephron and it is convoluted, which means it's coiled. The lining of proximal convoluted tubule is made up of epithelial cells which are joined end to end, pretty much like any single cell lining, lying on a basement membrane. And right across that basement membrane, you will have the peritubular capillaries. So the diagram that I'm drawing in front of you, it shows to you this very detail that this is the blood in the peritubular capillaries. This is the peritubular capillary. This one showed in red, that will have your blood. This is the single cell lining of the peritubular capillaries. Inside this region, which is inside the two green lines, this is the inside of the PCT, the proximal convoluted tubule. So if this green line, if I try to magnify this, it is made up of these kind of epithelial cells. The epithelial cells seem to be having extensive microvilli, which increase the surface area and which helps in allowing reabsorption to occur at a very fast rate. So this is the type of cell, you can say, display, or you can say how the fluids are separated out. This, you will have blood here, you will have tissue fluid here, and here you will have the filtrate which came from the Bowman's space. What happens further? The capillary endothelial cells can be seen. This is single cell lining. I told you this is the basement membrane which holds the lining together. Here you will have the tissue fluid filled. This entire width are your PCT epithelial cells, and the cells before it are of the peritubular capillary, and here you will have your tubular lumen where the filtrate is flowing that just came from the glomerulus, that came into the Bowman's space. Single cell lining of epithelial cells are held together in such a way that there are lateral spaces present between two adjacent epithelial cells. And here you have a tight junction between every adjacent epithelial cells of PCT. The tight junctions prevent the free mixing of filtrate with the tissue fluid. Therefore, the entry or the free passageway of solutes from here is prevented. So whatever is in the filtrate will not right away go into the tissue fluid or into the blood. It will have to cross the tight junction, or it will have to pass through the cell membrane of the PCT cells. Cations such as calcium and magnesium can cross the tight junctions. The PCT cell, as I told you, is having extensive microvilli. There are a lot of mitochondria involved here, and these mitochondria provide ATP for active transport, which is required for reabsorption. The two surfaces of the epithelial cells of the PCT are basolateral surface, also called as the basal surface. This is called as the basolateral surface facing the blood vessel. We also call the same surface as the basal surface or the basal membrane. Whereas the side of the cell facing the filtrate is called as the apical surface or the apical membrane. You can see that the apical membrane or the apical surface is having a lot more microvilli as compared to the basal surface. There is a sodium-potassium pump present on the basal surface that brings about exchange of sodium ion and potassium ion using active transport. How is this active transport occurring? This protein here, sodium-potassium pump, it allows 3 sodium ions to go out of the cell using active transport. So there is a low sodium concentration inside the cell. Still, more sodium keeps going out of the cell from the basolateral surface to the tissue fluid, and then it diffuses into the blood. Blood carries the sodium away. As a result, there is a low sodium concentration inside the epithelial cell. This low sodium concentration is established by breaking ATP into ADP and PI, and the energy is used to push the sodium ions against the concentration gradient. This is active transport or primary active transport, where the molecules or the substances are moved against the concentration gradient using energy from the ATP hydrolysis. The low sodium concentration inside the epithelial cells become a driving force for the functioning of the co-transporter proteins located on the apical surface. The apical surface is having a co-transporter called as sodium glucose co-transporter. This allows sodium and glucose to enter together through this carrier protein. Another co-transporter brings amino acid and sodium together. As a result, the amino acid and glucose, they both, once they have entered the epithelial cell, they go to the blood, they diffuse there. So you can see that in a similar way, inorganic phosphates also get reabsorbed into the blood. So there are various carrier proteins present on the apical surface. The more folded it will be having more co-transporter proteins. And all these co-transporter proteins, they work by moving one sodium ion and another solute together with that sodium ion. The basic driving force comes from the low sodium concentration established inside the epithelial cells by the working of the sodium-potassium pump, which is present on the basal surface and constantly sending out 3 sodium ions outside the cell and into the blood using energy from ATP. This pump's functioning is an example of primary active transport, as I already mentioned, whereas the functioning of the co-transporter protein is an example of a secondary active transport, which at times can be regarded as passive transport because it is not using energy of ATP directly. However, it's not scientifically correct to call this as passive transport. This is secondary active transport. Secondary active transport is the transport where the energy stored in the electrochemical gradient is used to pull a substance against its concentration. Inside the filtrate, you have a high sodium concentration, and here inside the epithelial cell, there's a low sodium concentration. The natural tendency of sodium ion is to enter inside the cell, so the electrochemical gradient favors the entry of sodium ions. The presence of these co-transporter proteins facilitates the process, and when it facilitates the process, sodium, when it's coming, it couples its movement either with glucose or with amino acids or with inorganic phosphate, bringing them inside the cell using that process. These solutes are then transported to the blood, which cause the blood to become high in solute concentration. As a result, water gets reabsorbed as well, and this water reabsorption is called as solvent drag. From filtrate, water goes into the blood. It just follows what the solute was doing. The filtrate, whatever is left of the filtrate, will then go to loop of Henle, where further reabsorption will take place, and all the reabsorbed solutes and nutrients, they will be added back to the blood and they will be taken back to the circulatory system. That's it from my side. Thank you so much.